So, hello Prem. Today we are going to study the entire human physiology, starting from respiration, circulation, excretion, locomotion, neural, and then chemical coordination. So, it's just going to be an 11th easy chapter because it's like an entire unit of human physiology containing all the easy chapters but still containing some important points that we usually forget. Oh, damn. Physiology. So let's just begin starting from respiration and just basically breeze through the entire unit. This will contain all the important lines and the important concepts that you need to know, while excluding all the easy concept and easy lines that we already always remember. This will contain only the important stuff that we most often forget. So first things first, what does our body do with oxygen? When we inhale, we breathe in the atmospheric oxygen. The oxygen enters our body and then it is diffused out through our alveoli and the capillaries, goes through the tissues, undergoes catabolism, and then the oxygen breaks down, carbon dioxide is formed. So what does the oxygen do in this context? Oxygen is basically there to break down the simple molecules, and simple molecules we have studied that in biomolecules can be amino acids, it can be glucose, it can be fatty acids, like these things are the simple molecules. So oxygen breaks down simple molecules is the important line in the first paragraph, whereas everything else is just boring and easy. Now let's go towards the next, which is the respiratory organs. So respiratory organs can be a little hard to remember. For example, so we will start with the lowest of forms. Like for example, we have sponges, cnidaria, flatworms. Like these lower organisms, we have lower organisms that basically undergo diffusion and throughout their entire body surface. So we will just go with body surface, whereas we have a little when we go higher in the hierarchy, we have arthropods. In that we have aquatic arthropods, we have terrestrial arthropods. In the case of aquatic arthropods, when we go for aquatic arthropods, we have gills called as bronchial respiration. In the case of annelids like earthworms, we have their moist cuticle. In the case of humans like us, chordates, we have, like in the end, let's just say humans, because in chordates we also have fishes that use their gills. We have lungs, and everybody knows that lungs is also called as pulmonary respiration. And then we have fishes that have their gills again. So just the simple stuff. Okay, and then we have some exceptions, like we have amphibians. We have frogs that can also respire through their moist skin, which is called as cutaneous respiration. This is again something that we have learnt always, simple stuff, nothing really important enough to write, because this is a line that we never really forget. Now when we move towards the human respiratory system, everybody knows about the nostrils, the nasal passage, nasal chamber, pharynx, larynx, trachea and everything. So but one thing which we might forget is that inside the trachea, which extends up to the mid-thoracic cavity, we have the primary bronchi called as a bronchus, and this bronchus divides into the first division into two bronchi, that is basically the primary bronchi. So the division of bronchus into bronchi is happens at the fifth vertebra. So this is important line. So first we had about this, then about this. Now the division of bronchus into the primary bronchi happens at the fifth vertebra. Which fifth vertebra? Thoracis. Fifth thoracic vertebra. And then some important lines can be starting from the trachea through the primary bronchi, secondary bronchi, tertiary bronchi, and then the initial bronchioles are all included, are all supported by incomplete cartilage. So if someone tells you or asks you that is bronchioles supported by incomplete cartilage, is going to be false. The initial bronchioles, yes. The final bronchiole that basically ends with the alveoli, no. So starting from the trachea till the initial bronchioles are all supported by incomplete cartilage. And then we have the double pleural membrane with the pleural fluid in between, which reduces friction. Now the outer pleural membrane is in contact with the thoracic lining, whereas the inner one is in contact with the lungs. So that's again pretty easy. And the lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The assertion reasoning question, where basically why does when the diaphragm contracts and when the external intercostal muscles contract, what happens is that the volume increases inside our chamber, inside our thoracic chamber. So since the volume increases, the pressure decreases, and since lungs is anatomically an airtight chamber, that is why that changes in the thoracic chamber reflects onto the lungs, and that is why the pulmonary volume increases and the pulmonary pressure decreases, and that is why the air is forced inside the body, and that is inspiration. So that is a very beautiful assertion reasoning. So let's just write the line that lungs anatomically is an airtight chamber, and I just told you the entire meaning behind it, and then everybody knows how the rest works. Now let's just talk about what contracts and what relaxes during inspiration and expiration. So in inspiration, the thing that contracts is the diaphragm and the external intercostal muscles, not the internal, not the entire intercostal muscles, but the external intercostal muscles. Okay, and then there is another called as the internal intercostal muscles, which we will talk about that in the expiration part. So these contract and they so basically in inspiration the diaphragm contracts and the external intercostal muscles contract, and this causes an increase in the pulmonary volume. But now what we have to understand here is the main line of the NCERT, that in the case of diaphragm is present on the posterior side of the body, so that is why they cause an increase in volume in the antero-posterior axis. Whereas since the external intercostal muscles are basically lifting up the ribs, they cause an increase in the dorsoventral axis. Okay, so this causes an increase in the thoracic chamber's volume, which basically reflects on the lungs, and that is why it causes an increase in the pulmonary volume, decreases the pulmonary pressure, and that is why the air rushes in. Simple as that. And now let's talk about expiration. So expiration will of course have the relaxation of the diaphragm, and one thing to note here is that in NCERT they have written the relaxation of the diaphragm, but along with that they have written the relaxation of the external and the internal intercostal muscles. which is an important difference, because in inspiration the only thing that is contracting are the external intercostal muscles, but in expiration both the external and the internal intercostal muscles are being relaxed. So this is basically contraction and this is relaxation. Let's move on. Yeah, we can just remember just a simple point that our body in a minute can breathe in and breathe out. Oh shit, what happened? Yeah, our body in a minute can breathe in and breathe out for a total number of 12 to 16 times per minute is the amount of time that we respire. And now we can jump on to the respiratory volumes and the capacities. So basically this is pretty easy stuff. Tidal volume, IRV, ERV, RV, IC, EC, FRC, VC and TLC. So basically you can understand the definition of what they mean by just what they are called. And the only thing you have to remember here is the formulas and the volumes. So that's the only important thing here. So in TV, tidal volume, it is 500 ml, which becomes around 6K to 8K per minute. Okay, and what does TB mean? Tidal volume is basically the amount of air that we respire or expire in a normal breathing. Now if we can go to the inspiratory reserve volume, is basically the additional volume that we breathe in after a forceful inspiration. This averages around 2500 to 3000 ml. Whereas the opposite is the ERV, and that is going to be 1000 to 1100 ml. Now the residual volume is going to be the amount of air that is remaining inside our body after a forcible expiration. As it says, residual means the amount which is remaining, and this is going to be just 1100 to 1200 ml. Now based on these volumes we can calculate the capacities, and the capacities, the first capacity is the inspiratory capacity IC, and then we have the expiratory capacity. For the inspiratory capacity is basically the amount of air that we can breathe in after a normal expiration, and vice versa for the expiratory capacity. So this will be TV plus IRV, whereas EC will be TV plus ERV. And then we have the functional residual capacity. Now what is the difference between the residual volume and the functional residual capacity? Functional means something which is realistic. So residual was the amount of air remaining after a forcible expiration. So functional residual capacity will mean the amount of air remaining after a normal expiration, because we are setting the realistic terms here. So this will be basically ERV plus RV. And then the next is vital capacity, and then the total lung capacity. The vital capacity is the amount of air that we can breathe in or breathe out after a forcible expiration or inspiration. This will be ERV, IRV and TV. Whereas total lung capacity is basically after a forcible inspiration, the total amount of volume of air that is remaining inside our lungs, not remaining, that is present inside our lungs, is the total lung capacity. So basically just plus all the volumes. So starting from TV, oh, how did the color change? So starting from TV to IRV to ERV to RV. So simple stuff. The definitions are pretty easy to understand. And then we have the exchange of gases. When exchange of gases is basically upon the pressure gradients, but there are also multiple things that can reflect on the amount of diffusion that takes place. So if I had to say the exchange of gases has three conditions, or the three factors that determine how much diffusion of gases is going to take place. The first are the gradients. the pressure gradients, the concentration gradients between the oxygen and carbon dioxide and other factors. The next we have is the solubility, and the next is the thickness of the membrane, because the thicker the membrane, the lesser diffusion will take place. Okay, and then we have some values of oxygen and carbon dioxide. We can just make that pretty easily. If this is PO2, this is PCO2, we can have the atmosphere, we can have inside the alveoli deoxygenated blood, oxygenated blood, and then we have the tissue. So let's just create it first. The amount of PO2 in the atmosphere is 159. If I remember correctly, we have in the alveoli a little less, I think 104. Yep. In the oxygenated blood it is around 90, 95. In the tissue and deoxygenated blood it's going to be the same, it is going to be 40, 40. So that's how you remember it. In the atmosphere it's the longest, it's basically the biggest, not the longest, it's basically the biggest, 159, and as it goes from the outside to the alveoli it decreases to 104, and then from 159 to 104, and then when it diffuses into the oxygenated blood it reduces to 95, and then after the tissue part the oxygen is catabolized, the carbon dioxide builds up, so oxygen is reduced to 40, 40. Whereas carbon dioxide is of course very low on the atmosphere. Now we need to understand a simple concept. Carbon dioxide diffuses from the deoxygenated blood to the alveoli, which means that the deoxygenated blood should have a higher value than the alveoli. So if deoxygenated blood has a value of 45, alveoli will have a value of 40. So that is why it is able to diffuse from deoxygenated blood to the alveoli. Okay, so the tissue will contain the same as that of the deoxygenated blood, whereas the oxygenated blood, oh, this thickness changed for some reason. Yeah, now it's good. So where were we? 40 on the alveoli, and tissue will have 45, and oxygenated blood will contain 40, and this is the entire chart of this. And one thing to remember here is that that these values that we have of PO2 and PCO2 are all based upon mmHg, millimeter mercury. Now we can jump on some more NCERT lines, which are the solubility of CO2 is 20 to 25 times greater than that of the solubility of oxygen. And then we have another important line which is going to be about the alveoli. So for example, if this is the alveoli, so we can just create this layer, and if this is a blood capillary that is going on, so we can create that in red actually. And if this is a substance in between, then always remember, when we are talking about diffusion, this will be squamous. This will be squamous epithelium. Whereas whenever there is capillaries, there is always endothelium. We will have the same thing on excretion also. This will be endothelium. Endothelium of capillaries, and the center will be the basement substance, which will contain the basement membrane. And if we had to write something more, we can say that the total thickness is much less than a millimeter, which basically means that the entire thing is basically almost less than even 1 millimeter. So that's again pretty interesting. We can move on to the transport of gases, oxygen and carbon dioxide. Important. So we have oxygen and we have carbon dioxide. Now we have to basically understand the distribution of oxygen and carbon dioxide. So the transport occurs by RBCs and plasma. That's it. So RBCs for oxygen carries 97% of the oxygen. If we divide, we can do this. So this will be 97% in the RBCs. This will be 3% in the dissolved state in the plasma. That means that the oxygen is directly dissolved into the plasma. In the case of carbon dioxide, it is kind of complex, because in carbon dioxide there are multiple phenomena which are not given in NCERT, like the Hamburger phenomenon thing. So we do not care about that, but basically it's that the RBCs and plasma contain small quantities of an enzyme called as carbonic anhydrase. I'm pretty sure, yeah, carbonic anhydrase. And by using this enzyme, the carbon dioxide basically binds with water to form bicarbonate H2CO3, and then forms HCO3 minus plus H plus, and it is basically in this form that it travels the most, and this occurs inside the plasma. So what I'm saying is, carbon dioxide has three things. First is has carbaminohemoglobin in the RBCs, which is 20 to 25%. We can say basically pure RBC. In the plasma, in the dissolved state, it is 6 to 8%, I'm pretty sure. Yeah, 7%. Now the next thing is as a bicarbonate ion. Here, the fun thing is that what actually this refers to that is the carbon dioxide finds the carbonic anhydrase in the RBCs, and then it uses that enzyme to form the bicarbonate ion, and then it shifts towards the plasma, and then it is transported inside the plasma as a bicarbonate ion. So we have to remember that this starts in the RBCs and then shifts towards the plasma, and this is 70%. So that is the distribution of the transport of this. So we can add another page to go towards the transport of oxygen. Now this is pretty, again, we can write here that some stuff about oxygen and some such stuff about carbon dioxide. For oxygen, it's that for every 100 ml of oxy blood contains 5 ml of oxygen, whereas in the case of carbon dioxide, every 100 ml of deoxy blood contains 4 ml of CO2. Okay, and now we have the factors. We have the sigmoid. In oxygen we have the sigmoid curve, that the oxyhemoglobin formation, oxyhemoglobin versus PO2 saturation, or we can basically just say PO2, gives us a graph of sigmoid curve. We do not have any curve given for this, and the factors for oxygen is high PO2, low PCO2, low H plus concentration, lower temperature, the pretty easy stuff. But in the case of carbon dioxide, it is a little more complex. As per carbon dioxide, it only depends upon two things, PCO2 and PO2. If the PO2 is high, PCO2 is low, then it will be less. If the PCO2 is high, PO2 is low, then it will be high. It's just that simple. So what will happen? There will be a diffusion of oxygen from the alveoli to the oxygenated blood, which will travel towards the tissue. Oxygen is spent, carbon dioxide is built up. Now carbon dioxide will take the enzyme from the RBCs, form the bicarbonate ion, and then enter the plasma, and then it will travel towards the deoxygenated blood and then the alveoli, and then in the alveoli it will again combine back into H2CO3 and then into CO2H2, and then it will get expelled out of the body while expiration. It's that simple. If we want, we can write the this CO2 plus H2O in the presence of carbon anhydrase forms H2CO3. Again, in the presence of carbon anhydrase forms HCO3 minus plus H plus. Okay, so final is the regulation of the respiration. For that, we of course have the medulla, we have the pons, and we also have a chemosensitive area. Yeah, let's understand these. So for regulation, we first have the medulla, which contains the respiratory rhythm center. We have the pons, which contains the pneumotaxic center. Which can basically regulate the respiratory rhythm center by reducing the duration of inspiration and thereby the respiratory rate. So this is for reducing. Inspiration. And then we have the chemosensitive area, which is basically present adjacent to the respiratory rhythm center, and it is also highly sensitive to CO2 and H plus ions. That's it. And then we have the disorders. For disorders, it's just basically some main key points for the questions. The first thing which we should remember in this line is wheezing is one of the symptoms, and that it causes the inflammation of bronchi and bronchioles. Okay, in the case of emphysema, first thing we should remember is about what does it target. It targets the alveolar walls, it degenerates them. It is a chronic disorder. First thing, it is chronic, attacks the alveolar wall, and caused due to smoking. And then the occupational disorders, we have fibrosis, ileosis due to some dust and particles. So that is very easy to remember. So this was basically a 20-minute revision containing all the important lines of respiration chapter. So now we can start with the next chapter of the human physiology, which is circulation. Circulation, again a very easy chapter, a little bigger than respiration but still pretty easy. First we have blood, which is a special fluid, the most commonly fluid by most of the higher organisms. We have the lymph tissue fluid. Now the blood contains the fluid matrix, plasma, and the formed elements. So let's just go towards the percentages because everything else is pretty easy. So basically blood contains as we just said, fluid matrix, plasma, and the formed elements, which is basically RBC, WBC, platelets. So let's talk about plasma. So plasma is going to take 55% of the blood, and it contains 90 to 92% of water and 6 to 8% of proteins. In proteins, it contains 3 proteins: fibrinogen, globulins, and albumins. Now once again, if you remember these names, you can remember their function. Fibrinogen, from the name we can understand it will help in coagulation. Globulins, from the name we can understand it will help in defense mechanism, fighting, phagocytosis. And the albumins we can understand from aldosterone, albumin, it will help basically for the osmotic balance of the body. And it also contains a lot of ions and everything, and the plasma without the clotting factor is called serum. Everybody knows that. We can move on to the formed elements. The formed elements, we have the first thing, which is a RBC, also called as erythrocytes. It is a devoid of nucleus, binucleate, biconcave. Everybody knows that. One thing which we might not remember is that every 100 ml of blood contains 12 to 16 grams of hemoglobin. And everybody knows the lifespan, 120 days, the graveyard, the formation in the blood, in the red bone marrow. Graveyard of RBCs is a spleen. Simple. We can go to the WBC, which is a little more complex because it has a lot of stuff. So we can also write the quantity here, which is 5 to 5.5 million per cubic millimeter. In the case of WBC, it is 6 to 8K per millimeter cube. But this does have two things: granulocytes and agranulocytes, which means just to have granules and to not have granules. In the case of having granules, we have neutrophils, basophils, and eosinophils, whereas in this we have lymphocytes and we have monocytes. So everybody has their percentages. It's my way to remember. One of the ways for me to remember basically is by a chapter of physics. There is a chapter in physics called as the Newton's law of motion, and if you also say that as NLM, and you can remember that in this way by saying NLM is the size range. So basically the from size, by the way, I mean the quantity. So for example, from N, that means the largest quantity is the neutrophils. So neutrophils is going to be around 60 to 65%, the highest. And then we have L, lymphocyte, 20 to 25%. And then we have the M, monocyte, which is 6 to 8%. Yeah. Okay. And then the least is going to be, I think, basophils. Basophils is 0.5 to 1%. And eosinophils is 2 to 3%. So just some ways to remember them by NLM. Very easy. Now what do? Neutrophils and monocytes are under basically phagocytose, the infections and everything, phagocytosis. We have basophils secreting heparin, serotonin, histamine. We have lymphocytes, which is for the immune responses. We all know that. Now we can move towards the platelets. Also called as thrombocytes. It comes from the cell fragments of the megakaryotes, and their quantity is 1.5 to 3.5 million. For some reason I always forget this. 1.5 to 3.5 million. And now excessive, if there is a reduction in lymph platelets, it can cause hemophilia, which is an excessive loss of blood from the body. Or we have the blood groups. We have the ABO blood grouping, pretty easy to be honest. We have the two antigens A and B. One thing which is important here is that some people that might not see that in the case of ABO blood grouping, the two antigens which are A and B comes from the RBCs, and the two antibodies in response to the antigens comes from the plasma. So the two antigens are antigen A and B, and the two antibodies are of course anti-A and anti-B. So a person with blood group A would have the antigen A and would have the antibody anti-B. So blood group B will have the antigen B and the antibody anti-A. A person with the blood group A and B will have the antigen A and B both and will have no antibodies. That is why it is a universal recipient, because it can take anybody's blood, whereas O will have no antigen and will have both the antibodies anti-A and anti-B. That is why it cannot take anyone's blood. It can give anybody its blood because it has no antigens, and that is how the entire system revolves. And then we have the RH blood grouping. In which one thing to remember is that the RH antigen, for example, the antigens are always present in the RBC, so this will also be present on the RBCs. And I've also completely mastered that fact of the RH negative mother and the RH positive fetus on the first pregnancy. While the parturition, some quantities of the RH positive blood of the fetus can enter the mother, which will create, so the mother will create antibodies. So the next time on a second pregnancy, those antibodies can leak into the baby and can cause jaundice anemia, which is called erythroblastosis fetalis. So what can you do? You can administer her anti-RH antibodies. This is, for some reason, this is one of the hardest thing, but for me it's pretty easy, so I'm not going to include this. Let's go towards the coagulation of the blood. In coagulation of the blood, I think we have been studying this for like 8th, 9th, 10th, 11th, right now 11th. So we all know how this works. Prothrombin to thrombin by thrombic kinase, and then fibrinogen to fibrin in the presence of thrombin, and then the whole vice versa works. So one thing which we should remember is that fibrin, which are the threads, what are they made up of? So the fibrins are made up of damaged formed elements of blood. Now we have the lymph. So in lymph we have two things. We have the tissue fluid and we have the lymph. In NCRT they are shown as the same thing, but it's different because basically they are the same thing, of course, but their name differs from where they are located. So as in the first case, for the tissue fluid, when there are capillaries and the water soluble components squeeze out between the cells with the protein left in the capillaries, so that water and the water soluble components that secrete out, so that's just come out of the cell, are called as a tissue fluid. Now once the lymphatic system drops these fluid into the major veins, then this is called as the lymph, which contains lymphocytes, antibodies and everything for the immune response of the body. And the important line I would say in this case scenario is going to be maybe about the fats, that in the lymph the fats are absorbed through the lymph in the lacteals present in the intestinal villi. So there are lacteals, maybe something present in the intestinal villi through which the fats are absorbed. Basic. Now we have the circulatory pathways, in which the open spaces are called as sinuses. Everybody knows what is open circulatory system and what is a closed circulatory system. And fishes have a 2-chambered heart, reptiles have a 3-chambered heart, crocodile have a incomplete 4-chambered heart, but in NCRT it's given as a 4-chambered heart. We have birds and mammals with 4-chambered heart, which is 2 atria, 2 ventricle. And how the circulation works in 2-chamber, it's basically that the oxygenated blood is oxygenated in the gills, and then just that simple thing. In a 3-chambered heart, the oxygenated and the deoxygenated blood mixes in the ventricle, and then it is basically an incomplete double circulation, whereas a 4-chambered heart will undergo double circulation. Now we can jump onto the human circulatory system. So heart, mesodermal origin, pericardium, it's all right. Atria, ventricles. So everybody remembers the fact that the one that separates the atrium is the interatrial septum, the one that separates the ventricles is the interventricular septum, and the one that separates the atria and the ventricle is called as the atrioventricular septum. But what are these things made up of is something which we might forget. So the interatrial, interventricular, and atrioventricular. So this is made up of a thin muscular wall, and this is made up of thick muscular wall, whereas this is a thick fibrous tissue. Okay, and then we have the SAN, the sinoatrial node. We have the AVN, atrioventricular node. The numbers 70 to 75 times a minute is how much the SAN generates the impulse. So the average we can take it as 72 beats per minute. So SAN generates it, passes on to the AVN. The AVN has AV bundles, and then the AV bundles passes through the atrioventricular septum and emerges on top of the interventricular septum, and that divides into the bundle of His and then ends with the Purkinje fibers, which basically goes throughout the entire ventricular musculature, and that is what happens throughout the entire thing. So we have these nodal tissues basically, which has the, in NCERT, if I had to say, they are written as specialized cardiac musculature called as a nodal tissue. Specialized, why? Because they're specialized for the function of generating an impulse. And then you have the cardiac cycle starting from the joint diastole, and then after the simultaneous contraction of the ventricles, the increase of the flow of blood. No. After the simultaneous contraction of the atrium, the left and the right atrium, there is an increase of the blood flow of 30%. That is important. That atrial contraction increases blood flow towards the ventricles by 30%, and then we know the entire pathway, and then the ventricles, and then the right ventricle will lead towards the pulmonary artery, whereas the left ventricle will lead towards the aorta, which are guarded by the semilunar valves and everything. So now we can jump towards the cardiac cycle. Here the one thing which we can forget can be that the cardiac cycle has a total duration of 0.8 seconds. The total volume of the blood is 5 liter, and the body has the ability to alter the stroke volume as well as the heart rate and thereby the cardiac output. So basically what this means is that the cardiac output, which is 5 liter as per NCERT and 5400 ml as per HSE, the cardiac output is basically stroke volume multiplied by heartbeat. In this, the stroke volume is 70 ml. Yep. The heartbeat can be 70 to 75 times per minute. When you multiply these, you get the cardiac output, which is 5 liter or 5 liter 4000 and 5 liter 400 ml to be exact. So this line basically refers to this, that you can increase or decrease the stroke volume, you can increase or decrease the heartbeat, and hence we have the ability to increase or decrease the cardiac output. The electrocardiograph, that is something which we can understand by a stick figure. So the multiple electrical leads, one on each wrist and one on the left ankle. So if we take this as a left ankle, one here, one here, one here, and multiple on the chest is where the electrical leads are applied, so that they can understand their EKG, which goes from P to T, PQRST, where P is the depolarization of the atria, the QRS is the depolarization of the ventricles, and T is the polarization of the ventricles. This is of great clinical significance, pretty easy. So what is this? This is the left ankle, one in each wrist and in multiple electrical leads. So this is, we can just say, ECG. Double circulation, yeah, this is something confusing, that the blood vessels, the arteries and the veins have 3 layers: tunica intima, media and externa, and they are each made up of different things. So that is something which we can always forget because it's confusing. Oh, that is not the spelling of artery. One minute. We have the intima, media, externa. Intima is made of squamous. By the way, we just remember this one thing in the respiration, that the capillaries had the endothelium and the alveoli had the epithelium. So once again, this will have the squamous endothelium, as we also studied in the respiration. The intima, the media, will be made up of smooth muscles and elastic fibers. And the externa would be made up of collagen fibers. Okay, so we have the tunica intima, media externa. Tunica intima made up of squamous endothelium, media made up of smooth muscles and elastic fibers, and the tunica externa made up of collagen fibers. And now one more thing is that the tunica intima is comparatively thinner in the veins, which basically means that veins are basically less narrower than arteries. Now we can talk about the hepatic portal system. Very annoying thing to remember. Which exists between the digestive tract and the liver, because it's, of course, hepatic gives us the liver part. We have the coronary system, which is basically the system for the heart. The regulation of the cardiac activity is completely done by the medulla oblongata, which is basically by the autonomous nervous system. We have the parasympathetic and the sympathetic, by which we can affect the cardiac output by affecting the stroke volume or the heartbeat. We also have adrenal medullary hormones, which can also increase the cardiac output. So regulation, medulla, ANS, and also adrenal medullary hormone. And from what they mean from here is basically epinephrine and norepinephrine, which is basically adrenaline. And we can end the chapters with some disorders. The first is hypertension. which is, if the normal is 120 over 90, this is 140 over 100. Normal is 120 over 80, then this is 140 over 90. So let's just write both. Like if it goes from 120 over 80 to 140 over 90, it is basically considered as hypertension, which can affect the kidney and heart. No, not kidney and heart, kidney and brain, if I remember correctly. Yeah, kidney and brain. We have the CAD. One of the most important and annoying thing is that CAD, always forget that whether it is atherosclerosis or atherosclerosis, which is, I am always confused with that. So it is not anthero but atherosclerosis, which is due to basically that blood supply to the heart is poor, and that is the result of CCFF, which basically means the deposition of calcium, cholesterol, fats and fibrous tissue. Which is causing the inefficient blood supply to the heart. The next is angina pectoris. Angina pectoris is acute chest pain. We all know that when not enough oxygen is reaching the heart. So we can remember CAD as not enough blood is reaching the heart, and angina as not enough oxygen reaching the heart. So if you have to write, let's just write it, because you never know what can happen. Not enough blood for heart. Angina called as angina pectoris, acute chest pain, not enough oxygen. It occurs in okay. The last thing, we have the heart failure. We have heart attack and cardiac arrest. Heart attack is when a muscle gets damaged. Cardiac arrest is when the heart stops beating, and the heart failure is when there is a congestive heart failure, also because one of the main symptoms is lungs. One of the main symptoms is the congestion of the lungs. That is why heart failure is also called as a congestion of heart. And this is when the heart is not pumping blood effectively. So that is pretty easy. And this ends circulation. Now we can start with the third chapter of human physiology. That will be excretion. So excretion is probably the most annoying chapter in human physiology. It contains like so many random information. So let's just first start with the three nitrogenous wastes are ammonia, urea, uric acid, and that is why we have ammonotelism, ureotelism and uricotelism. And the ones that exhibit them are also very random, so it's important to remember them. So first let's talk about ammono. In this we have bony fishes, aquatic amphibians and aquatic insects. As in ureo, we have, of course, us humans, mammals. We have terrestrial amphibians, and marine fishes, where of course in this case the ammonia is the most toxic, whereas uric acid is the least toxic. In urico we have birds, reptiles, snails and insects. So a normal insect will undergo eurycotelism, whereas an aquatic insect will undergo a monotelism. So a lot of confusing examples here. And then we have the types of organs. For example, platyhelminths have protonephridia as well as rotifers and everything, so we can write that here. Protonephridia for the platyhelminths, rotifers, some annelids, and cephalocodids. And then next we have nephridia. Nephridia is mainly used by annelids like earthworms and everything. And then everybody knows about amalficant tubules for the insects, the kidneys for us humans, and finally the hard one, which is going to be antennal glands slash green glands for the crustaceans like prawns. Oh no, not prawn. Damn, my mind be going somewhere crazy. So now after this, some basics start. We can start with the human excretory system, which contains of course consists of the kidney, and kidney has a lot of characteristics. So let's go through it all. First thing, where is our kidney present? Kidney is present from the last thoracic vertebra to the third lumbar vertebra. The size is going to be, the length is given as 10 to 12 centimeter, the breadth is given as 5 to 7 centimeter, and the thickness is given as 2 to 3 centimeter. The weight is given as 120 to 170 gram, and also an important line is that the center of the inner concave surface, this can be pretty confusing because in question they can put convex or something like that, so that can confuse us. So for the center of the hollow concave surface is where the hilum is present. And then we have the renal pelvis, the calyces, the medullary pyramids. Between them is cortex, that is called as a renal column or the column of Bertini. Nephrons, a total of 1 million nephrons. We have basically there are two parts in the nephron. One is the glomerulus and the other is the renal tubule, which contains the Bowman's capsule, contains the PCT, loop of Henle, DCT, collecting duct, and so on. Now we have studied this in standard 9th and 10th, so we don't have to go pretty much. It's basically the same thing in the NCERT, but one thing which is extra that we have not studied is that how is the vasa recta and all the peritubular capillary network is formed. It is formed due to the efferent arteriole. efferent arteriole, which emerges from the glomerulus, and basically it forms a capillary network around the renal tubule called as a peritubular capillaries, and that is how it is basically it comes towards the loop of Henle in a opposite directions and forms a vasa recta. That is why they have the counter current mechanism in the future, and that we will talk about. So this is the one that forms the peritubular capillaries, and this is the one that forms the vasa recta. Now we have the urine formation, in which everybody knows how urine is formed, but some things that we might forget is how much ml of blood is filtered by the kidneys per minute, and that is I was talking about urine formation. So in urine formation we have two values that we need to remember. The first value is the amount of blood that reaches the kidneys per minute. For the total blood that reaches the kidney per minute is 1100 to 1200 ml per minute reaches the kidneys, in which 125 ml of filtrate is formed per minute, and this 125 ml per minute is called as the GFR, the glomerular filtration rate. And now as we know, the volume of blood in our body is around 5 liter, and here every minute around 1 liter is reaching our kidneys. So we can say basically that 1/5 of our total body volume reaches the kidneys per minute. And now we are going to make a resemblance with the alveoli of respiration and this of the urine formation. In this case, it's about a new page. The glomerulus, Bowman's capsule, and the basement membrane. In this case, if we realize, glomerulus is nothing but a tuft of capillaries of afferent arteriole. Okay, so those are basically arteries which will basically form capillaries. It's just a tuft of capillaries, and we just learned in respiration as well as in circulation, capillaries contain endothelium. So glomerular blood vessels will be endothelium, whereas Bowman's capsule will have the epithelium, and then there will be a basement membrane in between them. So just like in the case of alveoli, where alveoli was the one which contained the squamous epithelium, there was a capillaries which contained the endothelium, and there was a basement membrane between, basement space. Just like that, the glomerular blood vessels contains the endothelium, the Bowman's capsule contains the epithelium, and there is a basement membrane in between. And now these epithelial cells are called as podocytes, which contains some pores called as slit pores or filtration slits, through which the plasma basically secreted out due to ultrafiltration. And one more thing is that if we go up, this 125 ml of filtrate per minute, right? So if there is 125 ml of filtrate per minute, so how much that would equate in a day is basically also given in NCERT, which comes out to 180 liters per day. And then here's another interesting fact is that we only pee out about 1.5 liter, about 1 to 1.5 liter of pee is that we pee out. But here it says that there is 180 liters formed, so which means that out of the 180 liter, 99% of the filtrate is reabsorbed, and only 1%, that is going to be 1 to 1.5 liter, actually passes through. And now we can go towards reabsorption the function of the tubules. Yeah. So we have an active process and we have a passive process. An active process is to reabsorb the glucose, amino acid and sodium ions, whereas a passive process is to absorb the nitrogenous waste. Like, of course, some of them are absorbed to maintain the osmolarity, but we just have to remember, if they ask whether absorbing the glucose, amino acids and sodium ions is an active or a passive process, it's an active process. And if they ask about nitrogenous waste, it's a passive process. Simple as that. Now we have PCT loops, Henel's loop and distal convoluted tubule. So if we talk about PCT, first thing that we need to remember is that it is made up of cuboidal brush border epithelium. It reabsorbs 70 to 80% of filtrate and it secretes small quantities of hydrogen ions and ammonia to basically maintain the osmolarity. And then we have the loop of Henel. In the loop of Henel is basically primarily responsible for the osmolarity. Like as the filtrate passes down from the descending limb, the filtrate concentrates, and then as it ascends, it basically gets diluted a little and everything. So that's the whole point. It doesn't really have any role in the process of reabsorption, whereas DCT undergoes conditional reabsorption of course water and Na plus, and it releases H plus ions and potassium ions and ammonia to maintain the osmolarity and the sodium potassium balance in blood. So the PCT secretes hydrogen ions and ammonia, whereas DCT secretes hydrogen ions, ammonia and potassium ions, and the loop of Henel has different functions. And then we have the collecting duct, which basically does the same thing. Like if it wants to make the urine more concentrated, it will absorb more water. It also releases some urea into the medullary interstitium fluid for the osmolarity and everything. And then we have the mechanism for the concentration of the filtrate, which is the entire process of how how basically the cycle works. If I had to say it in an easier way, if this is the Henel and this is the recta, vasa recta. What is that language? Here, in this case, while the filtrate is descending into the loop of Henel, it is permeable to water. That means the water is released out, whereas while it is ascending, it is permeable to electrolytes, so it is releasing NaCl. Now here is the thing that it releases is the one that a vasa recta accepts. Now for example, in the ascending limb, the loop of Henel is releasing Na plus. So vasa recta, while it is descending, so this is like this and this is like this. So while the loop of Henel is ascending, the vasa recta is descending, and while it is descending, it will take those Na plus and release water. It will take NaCl and release H2O, whereas in this case, when the loop of Henel is releasing water, it will take water and release NaCl. Okay, this is the entire counter current mechanism, and that is what happens while the loop of funnel is ascending. It takes some quantities of urea, and that is why it reaches the DCT and then the collecting duct, and that is why the collecting duct secretes some quantities of urea into the fluid, the medullary interstitium fluid, to maintain the osmolarity that was disturbed by the loop of Henle. And in this process, as we go from up to down, the concentration of urine increases by 4 times. And then we have the regulation of the hormone, the regulation of the kidney function, which is basically by the hormone feedback mechanism, in which we have a lot of things. One thing is the ADH that we talk about in the chemical coordination also, that if we are losing excessive loss of water, through our urine, the hypothalamus secretes ADH. So the first thing is the hypothalamus, which secretes the ADH or the vasopressin, which basically allows for more reabsorption and prevents excessive loss of water, which is basically called a diuresis. The condition is called a diuresis, excessive loss of water, which is prevented by ADH. And this is a constrictor, a vasoconstrictor, which means that it increases the GFR. The other thing that we have is JG apparatus, which is the juxtaglomerular apparatus. When the GFR goes down, it basically activates the JG cells to release renin. In this case, the JG cells release renin, which makes angiotensin 1 and then 2. Now this basically has two functions. The first thing is that it is a vasoconstrictor also, and the other thing is that it basically stimulates the release of aldosterone. And how does it do that? By stimulating the adrenal cortex, because adrenal cortex is the one that secretes the aldosterone, and aldosterone will help in reabsorption. The vasoconstrictor will increase the GFR and everything. So as you can clearly see, like the ADH is increasing the GFR, the JG apparatus is increasing the GFR. So there is someone that has to keep this in check, and that is we have the heart with the ANF, atrial natriuretic factor, which is a vasodilator and keeps everything in check. Now we can talk about some maturation, in which is basically the stretching of the bladder stimulates the stretch receptors, which sends the signal to the CNS, and the CNS sends the signal back to the through the motor neuron, which allows for the contraction of the urinary bladder and the relaxation of the urethral sphincter, which releases the urine. We talked about a 1 to 1.5 liter of urine. One thing to remember is that how much gram of urea is excreted out through our urine, and so that is 25 to 30 gram of urea is excreted out per day through our urine. And urine is very helpful because we can test that, analyze that for some kind of malfunctioning of the kidney or some kind of metabolic disorders. The role of the other organs in excretion, actually this is pretty important. Like, what is the role of urine analysis? One thing is to find metabolic disorders, and the other thing is to find malfunctioning of kidney. Okay, now we move on to the next thing, which is the role of other organs in excretion. For that we have the lungs, for the CO2 and water. We have the liver with that bilirubin, biliverdin, cholesterol, and degraded steroid hormones. And then the next is our sweat, which contains NaCl, urea and lactic acid. Then we have the sebaceous gland, which through their sebum, releases hydrocarbons, sterols and waxes. And finally, last but not the least, our saliva also contains some urea. So let's just divide them all. Nice. Now we can move towards the disorders. Disorders is actually pretty easy. Hemodialysis, if we have to understand the concept here, it's basically that if you have a blood that contains a lot of urea and hence you have uremia, you take that blood into a unit which is surrounded by cellophane paper, and that thing is surrounded by a fluid which has the same composition as the plasma. And now since that surrounding fluid does not have urea and your blood does have urea, it will be diffused into that fluid through the cellophane paper, and then you can bring your healthy blood back into your body. So while you are giving the blood away, you use heparin, which is an anticoagulant, while you add the healthy blood back in your body, you use an anti-heparin. So that way, when the blood comes back in, you will be able to coagulate once again, and then the wound closes and everything. So that's pretty easy. Renal failures and all are easy. One thing which might not be easy is the renal calculi, which is basically stone or oxalates, insoluble mass of crystallized salts oxalates, and this marks the end of the chapter of excretion. Start with the fourth chapter. Yeah, the fourth chapter of the human physiology, which is going to be locomotion. This is a pretty straightforward chapter where the first page just talks about that locomotion are the voluntary movements, and locomotion does not like some functions. Some organs like tentacles of the hydra can be for locomotion as well as for the capture of prey, so there can be multiple functions. So there's that. All locomotions are movements, but not all movements are locomotion. There are types of movements. We have the amoeboid movement, we have the ciliary movement, and we have the muscular movement. So that is kind of important. Like in the case of when I talk about amoeboid movement, one name that we have to remember is that macrophages are the one that exhibits these, but one thing that we always remember are the WBCs. WBCs, which are basically leukocytes, are the ones that show amoeboid movement. We already know that. We have learned that in chapter 10th, in standard 10th, diapetesis, like the squeezing through the capillaries wala. So WBCs are macrophages, where macrophages are the main thing you need to remember. And there is again something called as microfilaments in the cytoskeleton elements. We have the microfilaments, so that is also important. Cytoskeletal element. Okay, so that is amoeboid. In the case of ciliary, we just have like our entire body that has the tubular structures that are covered by ciliated epithelium. For example, in this we have the example of trachea. We have examples like the passage of ova. So these are some examples of the tubules, we can say the tubular structures that are covered by cilia. So that is why the trachea has cilia, which helps to basically trap the dust particles and only allow the nice air to move past, whereas the passage of ova, of course, the infundibulum and everything, they are also all covered by cilia, which allows the passage of ova. And then the muscular movements, we all know that, which requires our skeletal, muscular and neural system for the entire locomotion. And we have flagellar movement. We also have flagellar movement, which is basically sperm. Flagellar movement, sperm is something that we all know, but something that we might not remember is sponges exhibit flagellar movement, and euglena, which is protozoa, like euglena, also exhibit flagellar movement. So in amoeboid we have macrophages, WBCs and microfilaments from the cytoskeleton elements. For ciliary we have trachea and the passage of ova, and flagellar we have sponges, protozoa, and in that we have euglena, and we of course have the spermatozoa, but we all remember that. And some facts about muscles would be that muscles take 40 to 50% of our muscle weight, of our basically total weight. So if you are 100 kilograms, so 40 to 50 kilogram of that 100 kg comes from muscles. And then on the basis of location we have three types: skeletal, visceral, cardiac. Pretty easy, but we have an important line in the visceral muscles, that is, visceral muscles are located in the inner walls of hollow visceral organs, like our intestine. Intestine is a hollow organ because it is a tube, so it's hollow inside, and the inner side of that hollow organ is what we is that which is covered by the visceral muscles. So that is why the visceral muscles are present. And the cardiac muscles, we all know that they are striated and everything. Muscle bundle, fascicles covered, held together by fascia. Muscle fiber, okay, this is an important line, that our muscle fibers are syncytium. This basically means having multiple nucleus. And then we have myofibrils, actin, myosin. We all know that actin is the I band, myosin is the A band. Between I band we have the Z line, between A band we have the M line. The part of the myosin which is not partially overlapped by the actin is the H zone. Like these are some things that we never forget, but some things like which we can forget can be whether the microfilaments or the myofibrils are parallel or lateral to the longitudinal axis. So one thing to remember is that the myofibrils are parallel to longitudinal axis. And when we talk about something like the structure of a contractile protein like the actin, the myosin, so one thing to remember here is that, again, some lines like actin is made up of G actins, and a lot of G actins make up the F actin. So there are two F actin, the filamentous actin. The tropomyosin runs very close to it, whereas we have the troponin. Troponin and a subunit of troponin masks the active sites of actin so that the myosin cannot bind with it. So these are some things that we just never forget. But some things which we can forget is whether the troponin is attached to the tropomyosin or the F actin. So the troponin is attached to the tropomyosin. And one more thing which I would say is that the diagram of the myosin monomer, which is the meromyosin, which is the monomer of the myosin. So a lot of meromyosins will make up the myosin. So this contains like this. It's like a DNA kind of situation here. So like this, something like this would have a head and another head. What I want to emphasize here is the fact that they have two sides. One is here and one is here. So the site which is on the top is for the actin binding sites, whereas the sites which are down are for the ATPase activity. I want the size to be low. Okay, and then we have the mechanism of the muscle contraction, which is basically when the stimulus arises, it releases acetylcholine, stimulates the muscle, then there is a release of calcium ions. So the subunit of troponin, which was binding the actin's binding site, detaches from it and starts to go towards the calcium to bind with it. So this exposes the actin, and myosin, by hydrolysis of ATP into ADP and inorganic phosphate, utilizes that energy and binds with the actin, pulls it towards itself, and that is basically the cross bridge and everything. And that is basically how a muscle contracts. And in the actin is like the while contraction, myosin pulls the actins closer. So the sarcomere or the sarcomere, which is the distance between two successive Z line, shortens. The A band retains its length, whereas the A band is also contracted, so it also widens. So basically it's like just some common lines which we can understand by a simple thing, like if this is Z, 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 while they are contracting, these will come a little closer, and then this will be the total contraction. So what are these? These are the Z line. And the distance between two successive Z lines is called as a sarcomere. So when contraction occurs, so this is the maximum contraction. In this state, the sarcomere of course shortens, the A band retains its length, whereas the I band where was that line? And the I band also gets reduced. So the I band gets reduced, the sarcomere shortens, and the A band retains its length. Simple as that. And then we can jump on to the skeletal system. So for the skeletal system, we have bones and cartilage, we have the axial skeleton, and we have the appendicular skeleton. The axial is basically the longitudinal axis, like we have the skull, ribs, sternum, vertebral column, whereas the appendicular is like the limbs, limbs, which are basically our hands and legs and everything, along with the girdles that articulate with the axial skeleton, are all comprised in the appendicular one. So if I had to basically just differentiate first, I would just do it like we have 206 bones. We all know that in this 206 bones, the axial contains like 80 bones, and this contains like 126 bones. So now in this 80 bones, 22 comes from the skull, which is the facial and the cranial, all comprises of the 22, and in this 22 it is divided into 8 and 14, where 8 is the cranial and 14 is the facial. And then we have some ear ossicles, we have, yeah, so let's continue. I was at the differentiation of all the bones: 206, 80, 126, skull 22, 8 facial, 14, 8 cranial, 14 facial. And then we have 1 hyoid. Rather than making it like this, yeah, so we have this skull, the skull, we have the ear bones, ear ossicles, we have the hyoid bone, we have the vertebral column, we have the sternum, and we finally have the rib cage, the ribs. So in this entire classification, in the skulls we have 22 bones, in which cranial contains 8 bones and facial contains 14 bones. Ear ossicles, there are 6 bones, 3 in each ear. Hyoid, there is a single hyoid bone, U-shaped. There are 26 vertebral column, which can be divided into 7 cervical. We have the 12 thoraces, 5 lumbar, 1 coccygeal and 1 sacrum. This is cervical, this is thoraces, lumbar, sacrum, coccygeal. We have a single sternum and we have 24 ribs or 12 pairs. This should all equal out to 80 bones, or else life is a lie, because I haven't really counted them all. And when we talk about the 126 appendicular, it's pretty easy, because 60 bones comes from our arms, whereas the other 60 comes from the legs. And then we have the 6 for the girdles. In the arms we have humerus, radius, ulna, where we have a single humerus, a single, like 1 in each arm, so total 2 and 2. And then we have the carpals, metacarpal and the phalanges. There are a total of 5 metacarpals, so total 10, 14 phalanges, so total of 28. So just 1 minute, 17 phalanges plus 3 is 17, 17 plus 5, 22. So there are a total of 8 carpal each hand, so there is a 16 carpals. In the case of legs, we have the same kind of distribution probably. We have the femur, tibia, fibula, 2, 2, 2. Then we have tarsals, metatarsals, phalanges, where the metatarsals will be the same as 10, same as 28, whereas this will be a little less, this will be 14, because there is also the patella, our kneecap, which is 2. And in girdles we have 3 things: scapula, clavicle, and the causal bone. 2 scapula, 2 clavicle, 2 causal bone. Okay, so this should include the entire distribution of bones. And if I had to say some lines, I'll say that the cartilage between the vertebral column is the hyaline cartilage. We can say that the ribs is bicephalic because of the double articulation, dorsally by the vertebral column and ventrally by the sternum. In ribs, we can say that the first 7 ribs are the true ribs, the 8, 9, 10 are joined with the 7th rib and not with the sternum, and that is why they are the false ribs, also called as vertebrochondral ribs. And then the last pair, 11 and 12, are the floating ribs, because they don't even attach with the 7th, nor they attach with the sternum. And if I had to say something more, then I will say that the scapula location is the second and 7th thoracic vertebra. And now if I had to say one more thing about the articulation, for the articulation of the scapula with the arm, I can say that the scapula has elevation called as a spine, and then it continues as the acromion. So after scapula we have acromion, which has the glenoid cavity, which articulates with the humerus. So like this is the entire pathway. The scapula is like a triangular flat body which has a ridge called as a spine, and then it continues from there and projects out as the acromion, and the acromion has a cavity called as a glenoid cavity which articulates with the humerus. So that is the entire thing that we should know. And then we have the pelvic girdle. In the pelvic girdle we have the causal bone, which consists of 3 bones: ilium, ischium, and pubis, which and the point of the 3 bones fusion is called acetabulum, and acetabulum is the one which articulates with the thigh bone. So I can say that the causal contains the ilium, ischium, and pubis, and the point of their fusion is called as acetabulum, which articulates with the thigh bone. And the point of the fusion of the pelvic girdle, so we have the pelvic girdle for the left leg and for the right leg, and the point of their fusion is called as a pubic symphysis. So fusion of is called as the pubic symphysis. Now the last part are the joints and the disorders. The joints is pretty easy. We have the fibrous, cartilaginous, and synovial. Fibrous is for the facial and the cranial bones. Cartilage is for the, for example, vertebral column. We have the synovial joint. Synovial joint has very important stuff. It has some examples which is very hard to remember. First we have the ball and socket, which is for the humerus and pectoral girdle. we have the hinge joint for the knee, pivot joint for axis. No, let's not start with axis. Atlas and axis. This is a first and this is the second vertebra of the cervical vertebra. We have the gliding, for the carpals, and the saddle between carpal and metacarpal. Now, to end things off, we will have the disorders. We first have myasthenia gravis. Two important things: it is an autoimmune disorder, and next it has a problem in the neuromuscular junction. Whereas muscular dystrophy is a genetic disorder and it is the progressive degeneration. Then we have the tetany, which is pretty easy, rapid spasm. We have arthritis, inflammation of joints. One thing that I would say about osteoporosis is that it is caused due to low estrogen levels. Then you have gout, which is uric acid crystals. I will say by this we can conclude the chapter of locomotion. So now we will start with our fifth one, neural system. This is probably one of the easiest chapters. Let's just start. So let's first start with the fact that what is the reason for the neural coordination, and what is neural coordination? Neural coordination is when two or more organs complement each other's functions, and that is basically coordination, and it is done to maintain homeostasis. That is an important word, to maintain homeostasis, normal functioning of the body. We have the neural system, in which we have some things like hydra is a network of neurons. We have an insect has a brain, neuron, ganglia and everything, whereas we of course have a very advanced brain. But it is still something important which I might forget. So something like hydra will just have a network of neurons, whereas like an insect will be a little more advanced with a brain, ganglia, not ganglia, and the nerves, which are the neurons, will form neural tissue. Whereas of course vertebrates, we are very advanced. Now if I had to define and distribute everything, it will be the neural system is divided into CNS and PNS. CNS is the central nervous system, whereas PNS is the peripheral nervous system. Now in PNS we can have two types of nerve fibers. We can have afferent fibers whereas we have efferent fibers. Now the of course they have similar functions, which is to, so of course the afferent fibers and the efferent fibers have their functions, in which the afferent fibers transmits impulses from the tissues to the CNS. So we can just basically write to CNS, and this will be away from CNS. And now PNS is divided into two things: the autonomous nervous system and the somatic nervous system. This is for the voluntary involuntary, and we also have one more actually, which is a visceral nervous system, the VNS. The visceral nervous system basically consists of everything. You can think of nerves, fibers, ganglia, plexuses, and everything. And in the case of ANS, it is again divided into two parts, which is the sympathetic nervous system and the parasympathetic nervous system. This increases things, this decreases things. Basically, it's that simple. And then we can move on to the neuron. Neuron contains the dendrites, cell body, and the axon, where the dendrites and the cell body contain something called as a Nessels granules, which is made up of RNA or something, not given in NCERT, so it doesn't matter. And it goes, the impulses go from dendrites to the cell body to the axon. It's that. And then we can also say that what actually happens is the impulses reach the axon, and the axon ends as the synaptic knob. Synaptic knob contains synaptic vesicles, which contains the neurotransmitters like acetylcholine. So here we have three types of neurons on the basis of the number of dendrites and axons. Okay, that's cool. So one axon and two or more dendrites, one axon, one dendrite, only one axon. This is the case which is very complex, of course, when you have multiple dendrites. So this is something which can happen only in the brain. So this is cerebral cortex. This is going to be the retina of the eye. And something like having only a single axon is of course abnormal, so that this is something which happens in the embryonic development. Wait, what happened? Okay, and then we have the generation and the conduction of a nerve impulse, which is very important. So by the way, I forgot about this, that if you have one axon and two or more dendrites, that is multipolar, and if it's one axon one dendrite, that is bipolar, and this is unipolar. Now we can talk about the generation and the conduction of the nerve impulse. When the neurons are at the beginning stage, they are at the resting stage, which is their polarized state. During that stage is when the axonal membrane allows specific conditions. It is selectively permeable to potassium ions and impermeable to sodium ions and negative proteins. Now this what this basically means is that if I create a circle, this basically means that the sodium cannot enter in the potassium can enter, and the negative charged protein cannot exit. So this cannot happen, this cannot happen, this can happen. Now, because of this, since the negative charged protein cannot go outside, this creates a negative charge inside the cell, whereas since the sodium ions cannot come inside, this creates a positive charge outside the cell. Okay, and when a stimulus hits, the charge gets reversed and then the current flows from the positive to negative, and the negative and just like how current flows from a higher potential to a lower potential, and that is basically the entire thing. So when the stimulus is received, the sodium gets completely permeable to the this cell, and so the sodium just rushes in. So this creates the positive charge on the inside and the negative charge on the outside, and then the current flows. That's just how it goes. But that is only a short thing, and then it again there is a reversal of polarity and everything. So that is that. And then we have the transmission of nerve impulse, which has, as I already told you, synapse is basically the presynaptic membrane plus the synaptic cleft plus the postsynaptic membrane. Synaptic vesicles towards the membrane where they fuse with the plasma membrane and then release the neurotransmitters which binds with the receptors, and the effect is either excitatory or inhibitory. CNS, everybody knows about the Mennings, so no. Okay, so now let's just reach towards the final point, because that is now the main thinking, which is the forebrain. So forebrain is basically the cerebral cortex and then the cerebral medulla. The cerebral cortex is grey in color because of cell bodies, whereas cerebral medulla is white in color because of a high number of myelin shelled neurons. And here we have a number of functions. For example, the association area is responsible for the functions like memory, communication, and intersensory association. And then we have something like the thalamus, which is all about coordination. And then we have something like the hypothalamus, which is all about thirst, hunger, temperature, and everything. And then we have again something like the hypothalamus plus the hippocampus plus the amygdala. This is referred to as the limbic system, which is of course for the libido and everything. Then when we go towards the midbrain has the corpora quadrigemma cerebral aqueduct and everything, and it is situated between the thalamus hypothalamus and the pons of the midbrain. So if here is a thalamus slash hypothalamus and here we have the pons, then this is where the midbrain will be situated. And then we have the hindbrain, which contains the pons. Pons basically connects everything. It consists of fiber tracts that interconnect different regions of the brain. We have the pneumotaxic center that we talked about in the respiration. The medulla, of course, controls the respiration, cardiovascular reflexes, and gastric secretions. Cerebellum in the hindbrain has a very convoluted surface to allow more surface area. The brainstem forms the connections between the brain and the spinal cord. The brainstem concludes the midbrain, pon, and the medulla oblongata. So basically, not cerebellum. That's it. Midbrain, pons, and medulla oblongata, and this is the end of neural system. And now we're officially at the last chapter, which is chemical coordination. So neural was 5. The chemical coordination. Basically, the important points, if I shall say, are going to be that the neural system and the endocrine system jointly coordinate and regulate the physiological functions of the body. Nerve fibers do not innervate. If I had to say the three important keywords for hormones secreted by the endocrine gland are non-nutrient, intracellular messenger, and they are produced in trace amounts. Now we all know about the endocrine system. Let's first talk about the hypothalamus. Hypothalamus contains neurosecretory cells, which are also called as nuclei. And then we have the GnRH, which stimulates the anterior pituitary to release LH and FSH. We have the somatostatin from the hypothalamus, which inhibits the release of growth hormone. That is important. In hypothalamus, we have the somatostatin, which inhibits the release of growth hormone. And then as it passes through the axons and the portocirculatory system, it reaches the pituitary. And if I had to talk about the pituitary gland, it is present in a bony cavity called as sella turcica, and it is connected to the hypothalamus by a stalk. There are two parts in pituitary, that is the adenohypophysis and there is a neurohypophysis. Adenohypophysis consists of the pars distalis and the pars intermedia, where the pars intermedia is almost fused with the pars distalis. The pars distalis is called as the anterior pituitary, which secretes a lot of hormones like growth hormone, prolactin, thyroid stimulating hormone, adenocorticotropic hormone, luteinizing hormone, follicle stimulating hormone, whereas pars intermedia only secretes one hormone, which is important, that because that is something which we might forget, that pars intermedia secretes a hormone called as melanocyte stimulating hormone for the melanin containing cells. Now when I talk about neurohypophysis, which is under the direct control of the hypothalamus, there are two hormones, oxytocin and vasopressin. Vasopressin is for the reabsorption of water, whereas oxytocin is for multiple things like the contraction of the smooth muscle and the ejection of milk from the mammary gland. So these are some basic stuff that we should know, like the gigantism, dwarfism, acromegaly. Acromegaly has one important line, like something more common as disfigurement of the face is something which we will remember, but something which we might not remember is that the disease is hard to diagnose in the early stages and often goes undetected for many years until changes of the external features become noticeable. So if they ask this question, so now name the disease, this might get hard. So let's just keep that in mind. The ACTH releases steroid hormones, glucocorticoids, and if I had to say another thing, LH maintains the corpus luteum. Diabetes insipidus is caused when there is excessive water loss, which causes water loss and dehydration. That is called as diabetes insipidus. So that is important. Now we can move on to the pineal gland, which is for melatonin. Melatonin is the hormone for the sleep-wake cycle and everything. But one thing which is hard to remember is that the pineal gland is present at the dorsal side of forebrain. is present at the dorsal side of forebrain, and also there is the normal functions like the sleep wake cycle, body temperature, menstrual cycle, pigmentation and everything. But there are some also weird features which we might not remember, which are metabolism, and there is the defense capability. The thyroid gland contains follicles and stromal tissue. There is T4, T3, tetra iodothyronine, and the triiodothyronine. They present on the trachea, 2 lobes, one in each, connected by isthmus. There is hypothyroidism and there is hyperthyroidism. In hypothyroidism is caused by the deficiency of iodine, causes goiter, and hypothyroidism causes a lot of problems. For example, if there's a pregnant mother, the child can have cretinism, low IQ and everything, deaf mutism, whereas adult women can have irregular menstrual cycle. Hyperthyroidism can be caused by the cancer or the development of nodules on the thyroid glands. Exophthalmic goiter or Graves' disease is a form of hyperthyroidism in which there is an increased metabolic rate, enlargement of the thyroid gland, protrusion of the eyeballs, and weight loss. Okay, so this should be something which I should remember, but we can forget this, that exophthalmic goiter slash Graves' disease, which is a form of hyperthyroidism, causes increased basal metabolic rate, which causes the protrusion of eyeballs, weight loss, enlargement of the thyroid gland and everything. And then some important features of the thyroid gland, like what does it do, what does the hormone of the thyroid gland do. First things first, it controls the basal metabolic rate, RBC formation, and also maintains the water and the ionic balance. Cool. And there is TCT, which is also secreted by thyroid, which basically counteracts the PTH secreted by the parathyroid gland. The PTH is for the reabsorption of calcium, so basically increasing the calcium level. So that is why there is TCT to regulate the blood calcium levels. So we can decrease it. So it is a hypocalcemic hormone, whereas PTH is a hypercalcemic hormone. It is a peptide hormone. Okay, nothing really important. And then thymus, we have the location of thymus is very hard to remember. Thymus is located between lungs and the ventral side of aorta. Thymocytes, T lymphocytes, B lymphocytes, cell mediated humoral. Okay, that's so easy. Adrenal gland, adrenal medulla, adrenal cortex. Now here comes the Addison's disease. This is caused basically due to the problems in the carbohydrate metabolism. So this is a problem related to adrenal cortex. It is not functioning properly, which causes problems in carbohydrate metabolism, causing acute weakness. And then you have the adrenal medulla, epinephrine, non-epinephrine, adrenaline, catecholamines, emergency hormones. That's all right. Now the adrenal cortex has three layers: zona radicularis, zona fasciculata, and zona glomerulosa. So if I had to go from inner to outer, there is radicularis to fasciculata to glomerulosa. This is the inner, this is the outer, and this is of course the middle layer. Now when I talk about adrenal cortex, is what I talk about is corticoids. When I talk about adrenal cortex, I talk about corticoids, which can, if I'm talking about the carbohydrate metabolism, it is glucocorticoids, and if it's about the ionic balance, it is mineral corticoids. For this we have cortisol, for this we have aldosterone. Each with their specific functions. For example, glucocorticoids, in which cortisol, it's all about gluconeogenesis, lipolysis, proteolysis. So basically, what is the function? It is to metabolize the glucose, inhibiting the cellular uptake and utilization of amino acids. So basically, glucocorticoids have the function of breaking down carbohydrate to give energy. So that is why when they do not function well, which causes Addison's disease. So there, of course, their function is gluconeogenesis and also lipolysis, proteolysis, and then cortisol is also there for the cardiovascular system and kidney functions. Kidney functions, I think it is also there for the RBC production. Yes, it is. And inflammatory reactions. Oh, my bad. Not inflammatory, anti-inflammatory reactions. And then we have the aldosterone, which is for the reabsorption of Na plus and water. Along with that, yeah, phosphate ions and everything. Maintenance of electrolytes, body fluid volume, osmotic pressure and blood pressure. So that's pretty basic. So basically for Na plus, K plus, water and that stuff. Androgenic steroids are also secreted by the adrenal cortex, which is basically helpful for the growing of pubic hair during puberty and shit. Whereas pancreas, there are 1 to 2 million islets of Langerhans, and 1 to 2% of the total pancreatic tissue is islets of Langerhans. So 1 to 1 to glucagon, insulin glucagon, hyper glycemic, whereas insulin is a hypoglycemic. So glucagon affects the okay. So what did we say before this? Islets of Langerhans, which is 1 to 2% of the total pancreatic tissue. And now we have glucagon. Glucagon basically is present on the hepatocytes, which is the liver, and causes a lot of shit, whereas insulin keeps check on glucagon and is present on the hepatocytes also, as well as adipocytes. So here we have the diabetes mellitus. Diabetes mellitus is basically where the insulin is not working effectively, it's pretty low, so it causes hyperglycemia, and that too prolonged hyperglycemia, and it can cause ketone bodies, harmful compounds. So and it can be and it is curable by insulin therapy. And then we have the testes and the ovary. Testes, this is something which we have studied a lot in the human reproduction. Made of seminiferous tubules and stromal interstitial tissues, ovary and follicle stromal tissues. Yeah, something like this can be important, which is basically progesterone can stimulate the formation of the alveoli. So one thing which is there, the alveoli is formed because of progesterone. It forms the alveoli. Hormones of the heart, kidney and the gastrointestinal tract is important. Like for the heart we have the ANF, which is a vasodilator, and hence it decreases the blood sugar level. We studied that in excretion. And as per the kidney, juxtaglomerular cells, the JG cells, renin, we again studied that, which increases the GFR. And then there is also the erythropoietin. Okay. The juxtaglomerular oh lord, the spelling was crazy. Glomerular cells of kidney secretes erythropoietin, which is basically of course the formation of RBCs by erythropoiesis. And then we have the gastrointestinal tract with the gastrin, secretin, cholecystokinin and the gastric inhibitory peptide. So what does gastrin do? HCl and pepsinogen. Secretin is all about the bile juices. It acts on the exocrine. First things first, it acts on the exocrine pancreas and is helpful for water and bicarbonate ion. And then FCCK, cholecystokinin, and this one is present for that. Yeah, acts on the pancreas and the gallbladder. So this will be of course be utilized for the bile juice and the enzymes, pancreatic enzymes and bile juice as per that. And the GIP, the gastric inhibitory peptide, is there to of course inhibit gastric secretion and growth factors. Now, gastric secretion and motility. Oh lord, I'm tired. And now there are several other non-endocrine tissues called as the growth factors. These factors are essential for the normal growth of tissues and their repairing / regeneration. We have the mechanism of hormone action. The hormone binds with the receptors, forms the hormone receptor complex. The hormone either binds with the membrane or either goes to inside the cell and binds with the nucleus. Depends upon what kind of hormone is being released, which causes some kind of metabolism, and hence a physiological function are regulated by hormones that we studied before only on the first page. There are peptide, polypeptides, protein hormones like insulin, glucagon, pituitary hormones, hypothalamic hormone, etc., There are steroid hormone like the estrogen and progesterone, testosterone, and everything. Iodothyronine, we studied the thyroid hormones. Amino acid derivatives like epinephrine. Yeah, this is important. I'll say the last section would be about this, that in the case of protein mechanism, in the protein hormones, the hormone binds with the receptor present on the membrane. This causes the release of the second messenger. Examples: IP3, cyclic AMP, calcium and everything, which will just basically regulate the functions. For example, the physiological and the okay. And then the other is a steroid one. Steroid iodothyronines follow this mechanism where they basically, the hormone goes inside the cell and binds with nucleus. So an example of this would be, first things first, this would be the ovarian growth, whereas the example of this would be something like tissue growth and differentiation. For example, when the estrogen enters the uterus to proliferate the endometrium, like this can be an example. And with this, the human physiology is over completely.